Thanks, Sean. So first off, I'd like to take this opportunity to thank you all for attending this important event. It's a key day for us at the Robarts Research Institute, as well as at the Schulich School of Medicine and Dentistry. Today's a milestone on several different fronts for us as an institute and as a school. At the beginning of this, we're going to be welcoming back to the campus a number of individuals who are absolutely critical with regards to their vision for the establishing of the Robarts Research Institute. And as you know, this is the 25th anniversary of the founding of the Institute. So today, you'll be seeing a number of individuals who I think you'd be really wise, if you have, particularly students, if you have a few minutes to go up and say hello to. You'll get a tremendous insight as to how the Institute was founded, their visions as to what science would be. So at varying points during the course of the day, you'll be seeing either Dr. Stiller or Dr. Barnett, Dr. Gunton, Mr. Ivey or Mr. Blewett amongst the crowd or at some of the events. And by all means, go up and say hello and ask them, why did you do this? This evening we'll be celebrating not only them, but a number of their colleagues who unfortunately no longer with us, Dr. Drake, Mr. Blackburn and Mr. Taylor, after whom the symposium is named, as the founders of the Robarts, and that'll be part of our festivities this evening. We'll also be marking today a significant change in our vision for what this particular day means to us as a school. I think by now all of you will be aware that our guest speaker for this evening is Michael J. Fox. I've rapidly discovered during the day that you can actually age an individual not by the number of rings around their legs, so to speak, as a tree, but rather what part of his career you actually identify yourself with. So I'm not going to tell you what part I identify myself with, but I actually heard somebody who was really quite linked to Stuart Little earlier this morning for it, so I think there's a bit of a gap there in aging. But this really marks for us a seminal change in how we view this day and the celebration tonight, and even the dinner itself is the Leaders in Innovation evening. In the Schulich School, we'll be having two events in the course of the year that will celebrate what we do as a school. So the one will be the Gala Homecoming event, the first one of which was held to celebrate the 130th anniversary of our school. That will always be on the Friday evenings. It will always be a major event to bring back all of our alumni and students and current faculty into one venue to celebrate the school. This will be the second. This event will celebrate our academic achievements and our science, both as a school, but particularly within the Robarts Research Institute. And you'll see this expand over time. So bringing Michael here this evening as a speaker, somebody of that caliber and international stature, and his dedication to research, and particularly for Parkinson's disease, and his foundation, really states that we're going to do things a little differently. This is going to be a major day for us. So those will be the two in the course of the year. Today we're also going to be celebrating excellence in stem cell research. And so for those of us with less hair and more along the gray lines for it, when we were doing our training, stem cells was just a glint off in somebody's eye for it. And even in the late 80s, 90s, this was really something that was just starting to be a discussion. But well, there were stem cells, and who would have thought that you actually could take stem cells from an adult brain in the hippocampal region and actually demonstrate they still have the capacity to divide, and then to move on and understand you could take them from skin and elsewhere, and then that you could actually differentiate them into tools that we as investigators can use to ask very fundamental biological questions about cell differentiation and lineage that we could only have dreamed of 2 decades ago, 2.5 decades ago. And then to start to ask the question, how do you apply those? How do you move them into the clinical realm? And I think that's where we're going to see major advances, and already are. Today we're lucky. We have some of Canada's best, and internationally one of the world's best speakers, who you'll be hearing later on today, who's actually the recipient of the J. Allen Taylor Prize, and that will be Dr. Rudolf Janus, who will be speaking to us later at the end of the day. So for those of you who are students who are attending, and we recognize this as a constant flux throughout the course of the day, you're going to have an opportunity to sample some of the best and the brightest in the field of stem cell biology and to ask questions about how does this apply to what we want to be moving forward. I can't think of a more fitting day to talk about revisioning what we do as an institute and what we do with this particular award. Now finally, I also will be turning this over to Dr. Ravi Menon to introduce and make a few comments on behalf of the Robarts, but I also want to recognize Ravi, who as of today will be stepping down as the Associate Director External for Robarts. Ravi has shepherded these events over the course of the last several years and done so extremely well. And they've increasingly become more and more popular events with a greater number of attendees to that. This is not a trivial thing to be doing amongst all the other aspects of what he does with his research and also promoting the Institute. And for that, I think we all owe Ravi a deep debt of gratitude, and certainly my appreciation for that. And without any further words, Ravi.
So I'll just say a few words as to the genesis of the prize and this particular symposium. This is the 27th awarding of the Taylor Prize. But of course, as Mike just mentioned, it's the 25th anniversary of the Robarts Research Institute. And you might wonder how that could be, and we're actually not sure. So we're not sure when the symposia originally started, but certainly over the 17 years that I have been at the Institute, the way this has worked is the scientists, the leadership within the Institute every year has identified an area of endeavor within the Institute that we would like to recognize on an international basis. So we strike an internal committee which will get calls for nominations from basically all over the world. This is a huge deal. Hundreds and hundreds of letters go out to deans of medical schools and biology schools all over the world seeking these nominations. The nominations come in and are first looked at by an internal committee which specializes in the area, except for me, who knows nothing about anything. But we had real stem cell biologists involved. And we make a short list. And after we make that short list, we avail ourselves upon a number of external external reviewers. And afterwards, we ask them to speak in our symposium as well. And so we are truly blessed in Canada because we have some of the world's most outstanding stem cell biologists literally within a stone's throw of London, Ontario. And they are here today to speak to you in this symposium. So I hope you enjoy the symposium. I hope you enjoy the day. And I hope many of you will be able to make it to the dinner tonight. And with that, I'm going to ask Derek van der Kooy to come up and Sean to introduce him.
So as Ravi mentioned, we're privileged to have with us today several internationally recognized stem cell researchers that are going to share with us some of their exciting research findings. And our first speaker this morning is going to be Dr. Derek van der Kooy, professor at the Department of Molecular Genetics, University of Toronto. Didn't get a lot of background information for Derek, so I asked him what we should introduce him as a title, and he said just brilliant and handsome would be fine. So we'll go with that. But Derek's going to share with us today his work on pancreas-derived multipotent precursor cells and their application in vivo for the treatment of diseases such as diabetes. So with that, thank you, Derek.
Thanks very much for the planted introduction. And thanks also for the invitation to allow us to help celebrate the Robarts Day, as well as this year's winner of the Taylor Prize. So I think we don't need to turn them on. I'm going to talk a little bit about evolution and an idea that the pancreas actually evolved from the brain. And this is an old idea. More than 50 years old. It started with people who have compared the biochemical similarities of the brain and the pancreas. And these are people like Pierce, who put forward a theory based on the biochemical similarities between brain and pancreas. Later on, other theorists called some endocrine cells paraneurons, neuron-like cells. More recently, this has become even more of an interesting parallel, because it turns out many of the genes that are important for the embryonic development of the pancreas are also genes that are important for the embryonic development of the brain. And in fact, there's a whole slew of genes, among them neurogenin-3, PAX3 neuro-D, that are important for both the development of the brain and the development of the pancreas. So based on not much more than those similarities, we wondered, could we use a technique that had been very useful for isolating and studying adult brain stem cells, and use that same identical technique to see if we could isolate stem cells from the adult pancreas? And the answer was, yes, we could. So for almost 20 years now, we and many other labs have been using a very simple assay for studying adult brain stem cells, embryonic and adult brain stem cells. And this is the Neurosphere assay that was developed originally by our collaborators Sam Weiss and Brent Reynolds. And in this assay, you isolate single cells from the remnant of the embryonic germinals out of the brain, and those single cells, which we call stem cells, will proliferate in serum-free media in the presence of two growth factors, EGF or FGF2, and proliferate and make big floating spheres that float in the dish. And the reason why we call those spheres the product of the clonal proliferation of a single brain stem cell is that they show the two cardinal properties of stem cells, self-renewal and multipotentiality. Self-renewal is shown by the fact that those clonal spheres can be dissociated into single cells, and a small number of the cells in the spheres will make secondary spheres clonally, and you can do this repeatedly. The process, or the feature of multipotentiality was illustrated by taking one of those spheres and plating it down in a sticky substrate, adding some serum, and it turns out that every sphere that comes from the proliferation of a single brain stem cell will make neurons, astrocytes, and oligodendrocytes, all the types of cells in the brain. So we wondered, could that same simple serum-free assay for isolating brain stem cells be used to isolate a stem cell from the adult pancreas? And the answer was yes. And this is work that was done originally by two students in the lab, Margot Arnfield, a current PhD student, and Simon Smuckler, a PhD student who finished about a year ago. Okay, so what we did was dissociate adult pancreas into single cells from a mouse and grow them in serum-free medium in EGF and FGF, and what you found is that a very small number of cells, around 1 or 2 in 10,000, of the cells in the adult pancreas will grow spheres in a dish. And you can see that this is after 7 to 10 days. A single cell would make a colony of about 3,000 or 4,000 cells. If you take a single cell from the adult brain from the same mouse and grow under exactly the same conditions, you'll also get a sphere. But these scale bars are the same. In both cases, they're about 50 micron scale bars. So this sphere is much bigger, about 3 or 4 times bigger. It contains approximately 10,000 cells after a week or 10 days. Okay? So pancreatic cells make spheres, and brain stem cells make spheres. But these are rare cells. In the case of the pancreas, it's 1 or 2 in 10,000 cells. Now, you can also get these from adult human pancreas. So from adult human pancreas that are taken out in preparation for transplants of islets into type 1 diabetics, some of those islets are not good enough for transplant, and then we get them. And so if you dissociate adult pancreatic cells into single cells, a small number of those cells in serum-free media and FGF and EGF will proliferate to form spheres. And these spheres will passage. They're about the same frequency, actually. Again, a few in 10,000 cells. But if you take them and dissociate these spheres into single cells, a small number of the cells will form secondary spheres. Some of these will form tertiary spheres. And you can passage these cells for quite a long period of time. In fact, the human pancreatic stem cells passage better than do the mouse ones, which is a surprise for us. Now, these cells show some self-renewal, but they also differentiate. And when you differentiate them from either a mouse or a human pancreas, then those cells, those single pancreatic stem cells, will differentiate into all the different types of cells of the pancreas. So all the endocrine cell types, the insulin-positive cells that express insulin, GLUT2, C-peptide, they'll make glucagon-positive cells, somatostatin-positive cells, all the endocrine cell types. They'll also make acinar cells, which are the cells of the exocrine pancreas that excrete digestive enzymes. Now, besides all of the pancreatic cell types, they make one other cell type. They make a few neural cells. And you can actually see neurons that are coming out of single adult human pancreatic stem cell colonies. So this is really unusual, right? This is a cell from the adult pancreas that comes from the endoderm, germ layer in the embryo, that's making pancreatic cell types and is also making large numbers of neurons. And this is true of both adult mouse and adult pancreatic stem cells. Very unusual. Now, it depends, this wholly depends on these being clonal derivatives because, of course, there are lots of other neural cells from the neural crest that are in the vicinity of the pancreas. And so we want to make sure that these are actually clonally derived, and both with the mouse and human, we've done experiments where you mix together pancreatic cells from different color mice, red and green, put them in the same, and culture them together, and culture at a density where we see all the spheres are either all green or all red. And when under those conditions, we still find that single pancreatic stem cell colonies come from a single cell, will make all the cells of the pancreas and also make neurons. We've also plated single cells per well, over 15,000 wells, and a small number, 3 or 4 of those wells will give rise to colonies. Okay, so if you compare an adult mouse pancreatic stem cell with an adult mouse brain stem cell, grown as a single clonal sphere and differentiated, and compare what they differentiate. Now these, from the same mouse, an adult pancreatic and an adult brain stem cell were grown under identical conditions, were differentiated under identical conditions, and they produce different cell types. If you take a brain stem cell, they make a small number of neurons, 4%, make huge numbers of astrocytes, and basically that's it, astrocytes and oligodendrocytes. If you take the pancreatic stem cells, here we're calling them PMPs, pancreatic multipotential progenitors, because Simon liked saying the word pimps. I just call them pancreatic stem cells now. The shocking thing is that these pancreatic stem cells, single cells, make colonies where 25 or 26% of the cells are neurons that are differentiating from these things. They make very small numbers of glial cells, but they make all of the endocrine cell types, the insulin-positive glucagon and somatostatin cells, and they also make amylase-positive acinar cells. So you've got a cell from the adult pancreas that makes all pancreatic cell types and neurons, especially, and a brain stem cell type that only makes brain stem cells and nothing else. So how can this be? Well, so we wondered about what was the origin of the pancreatic stem cells, and we looked at the pancreatic stem cell colonies to see if they had markers of other lineages. So we looked for general endoderm markers, not specific for the pancreas. They weren't expressed. It wasn't a general endoderm cell. We look for pluripotency markers, markers that are expressed in the early blastocyst, and in embryonic stem cells, they're not expressed in pancreatic stem cell colonies as well. Look for mesoderm markers, they're not expressed. We also looked for neural crest markers. The neural crest, because these pancreatic stem cell colonies make neurons, we wondered, could they be taking origin from a neural crest cell? The neural crest arises in the embryo from the border between the neural tube as it foams up and the surface ectoderm. And the neural crest migrates everywhere in the body, essentially. And we wondered, and certainly you can see neural crest cells that have invaded the pancreas, and we wondered, could these cells actually not be derived from the endoderm, like the pancreas is, but from the neural crest? So we looked at a bunch of neural crest markers, and they express a couple of them, but they're not specific markers. So we decided to do lineage tracing, and we used a Wnt1 Cree mouse. This is an animal that expresses Cree recombinase behind the Wnt1 promoter. Wnt1 is a gene that's expressed in the early neural crest. We crossed it to a fluorescent reporter mice, such that any cell that expresses Wnt will turn green or yellow, depending on the reporter, and every cell in that lineage, all of its descendants, will similarly be green or yellow. And so this is a way to trace the neural crest lineage. And when we do this, we see in the pancreas, in vivo in the adult mouse, there are lots of neural crest cells surrounding the islets and within the pancreas. But when we dissociate that and grow spheres, clonal spheres from that dissociated pancreas, where the neural crest has been labeled, only 5% at most of those spheres come from neural crest. The other 95% do not look like neural crest cells. We also used another Cree recombinase animal where PDX1, the PDX1 promoter, is driving Cree recombinase. PDX1 is the name of a gene, pancreatic duodenum homeobox containing gene number 1. That's probably the earliest marker of the part of the endoderm that's going to give rise to the duodenum, which is the first part of the gut tube past the stomach, and the pancreas. So it marks in the embryo very clearly those two organs and nothing else. And if you use PDX1 Cree crossed to reporter mice, it turns out that 90% or close to 95% of all the pancreatic stem cell colonies come from PDX1 lineage cells. So what that means is the vast majority of these pancreatic stem cells that are making all the pancreatic cell types and neurons take origin from an endoderm derivative in the pancreas. So we looked in a little bit more detail, who are these rare pancreatic stem cells in the adult pancreas? And it turns out they're insulin-expressing cells. The stem cells themselves are insulin-expressing cells. And the way we showed this is to take mice that express green fluorescent protein behind the mouse insulin promoter. So in those animals, only insulin-positive cells are green. If you FACS sort the insulin-positive cells from the pancreas versus all the other ones, basically all of the spheres come from insulin-positive cells. Now, there are a special type of insulin-positive cells. Most of the insulin-positive beta cells in the pancreas express high levels of insulin like these cells do, but they also express high levels of the glucose-2 transporter that allows glucose to get inside cells. These stem cells, the rare stem cell population, expresses high levels of insulin, but very low levels of the glucose-2 transporter. That's more like an immature beta cell. So one of the ways we've been able to purify these cells is to use high insulin but low GLUT2 staining. So they're different from most of the beta cells that are present in the pancreas. So this is a sphere that was grown from a single mouse insulin promoter GFP pancreatic stem cell, and you can see the sphere, and there's lots of green cells that are insulin-positive cells. If you differentiate them in serum on a sticky substrate, you find that some of the cells will differentiate just like pancreatic endocrine cells, like beta cells. They have dense core vesicles, and they look just like, for all intents and purposes at the EM level, just like a mature differentiated beta cell from the mouse pancreas. These colonies also make separate insulin and somatostatin cells, glucagon cells, and separate insulin and glucagon cells. And these single colonies that come from single insulin-positive pancreatic stem cells make insulin-positive cells, and they also, as you can see, make nice red neurons with nice big processes. Okay. So we thought that perhaps the making of neurons by adult pancreatic stem cells was sort of a culture artifact. It's something that happened in culture because we're culturing under the same conditions that you culture brain stem cells. We're differentiating them under the same conditions that you differentiate brain stem cells. Maybe it's just something that happens in culture and doesn't happen in vivo. But we took advantage of a mouse that was made by and generously donated by Doug Melton, where this animal expressed a tamoxifen-inducible rat insulin promoter Kooy recombinase. And what that means is you can give a drug to the adult animal, activate Kooy recombinase only in cells that express the insulin promoter, and cross that to a fluorescent reporter, and that allows you to label in the adult, in a drug-inducible way, the insulin-positive cells. And remember I told you all of the stem cells, or vast majority, are insulin-positive cells in the adult pancreas. So if you do that experiment, and we gave 3 injections of tamoxifen, and with 3 injections of tamoxifen that doesn't become toxic, you can label 30 to 40% of the cells in the adult pancreas, and sure enough, we can label about 30 to 40% of the spheres in vitro that come from the proliferation of a single cell. But if you do that experiment in vivo, where you give 3 injections of tamoxifen, and then sacrifice the animal on day 6 after the end of 3 injections spaced by 2 days, what you find is 99.6% of all of the cells that are labeled with green fluorescent, that means they are insulin lineage cells, also express insulin as marked by insulin antibody staining. So this just says that yes, our reporter is faithfully marking cells that are expressing insulin. But if you let those animals survive for 2 months, what you find is the insulin lineage cells, the green cells, 15% of them are no longer expressing insulin protein. What are those other 15% of the cells doing? Well, some of them, so here now you're marked some insulin-positive, insulin lineage cells, some of them are now expressing pancreatic hormones, glucagon or somatostatin, some of them are expressing amylase, and some of them are expressing neuronal markers. So this means in vivo, in the adult pancreas, the pancreatic stem cells are making, not many, but they're making other types of non-insulin positive cells. So the stem cells seem to be functioning in vivo as stem cells to produce more than one type of cell. Most interestingly, these cells are making small numbers of neurons in vivo, adult pancreatic stem cells. So how does this happen? We've suggested that this is not a developmental problem. It's not that the neural crest is migrating down into the pancreas. It does migrate down into the pancreas, and it's also making many of the neurons in the adult pancreas. But some of the neurons in the adult pancreas are made intrinsically from pancreatic stem cells in the endoderm-derived pancreas. So how could this happen? Well, rather than suggesting a developmental origin of the neurons in the adult pancreas, we're suggesting an evolutionary origin. How might this happen? Well, one of the interesting things is that insulin has been around forever. It's expressed by single-cell organisms like E. coli. They express insulin. If you look at early multicellular organisms, they use insulin, but first they seem to use it, at least the animals that exist now that might be representatives of early multicellular organisms, express insulin in neurons. But these are animals that don't even have a pancreas yet. They have neurons, and they use insulin as a neurotransmitter. So insulin is used widely as a neurotransmitter in the animal kingdom. And what we suggest might have happened is that with the evolution of more complex multicellular organisms, something that gets used as a neurotransmitter in the brain, a communication molecule, also gets expressed in the gut in a cell that's going to become the beta cell of the pancreas. So how might this happen? Well, you can imagine if there's a master gene for for turning on a neuronal program, a mutation that allows that neuron master gene to be expressed in the gut could allow expression of a whole slew of neural genes in a cell in the gut, which could be, in principle, the origin of a pancreas. So any evidence that this could possibly be true? Well, again, if you look through organisms that exist now, you see a wide variety of cells that use insulin. So in E. coli and tetrahymena single-celled organisms, they express insulin. If you look at jellyfish, which are thought to be representative of one of the earliest multicellular organisms, they have nervous system where insulin is used as a neurotransmitter, but they don't have a pancreas. So insulin is expressed in the neurons, but there is no pancreas yet. Others, mollusks and crustaceans, have insulin expressed in the pancreas, but not in any cells in the brain, none in neurons. Drosophila is sort of interesting because all of the insulin in Drosophila comes from neurons. There is no pancreas. All of the insulin is released from neurons in the brain, in the Drosophila. So the idea is that insulin was first used as a communication molecule, and particularly was used in early multicellular organisms as a neurotransmitter to communicate between neurons. With continued evolution of multicellular organisms, a neuronal gene expression pattern, including insulin and other neurotransmitters, got expressed in the gut. And that was, by this model, the origin of the pancreas. Okay, so I want to just end by talking about a couple of recent studies that have been done by a new postdoc in the lab, Irina Brockman. And what Irina's asked about is, what are pancreas-derived neurons actually doing in the pancreas? Most of the neurons in the pancreas come from the neural crest, but a few of them seem to come from the pancreas. And so she looked at the PDX1 lineage cells, PDX1 Kooy crossed to a fluorescent reporter, looked at the cells in and around the pancreas. And one of the things she found is that if she looked in the gut, she saw some interesting locations of PDX1 positive cells. So these are PDX1 Kooy YFP. So these are adult sections through this is the duodenum. And you can see here with PDX1, I mentioned is a pancreatic duodenum homeobox-containing gene. It marks all the early cells in the endoderm that are going to give rise to duodenum and pancreas. And here's a loop of the duodenum. It's completely stained with the reporter. If you use Wnt1 as a marker for the neural crest, it, of course, doesn't stain the intestinal epithelium. But surprisingly, both PDX1 and Wnt1 independently stain the external muscle layer that surrounds the gut. Okay. And what's in this layer? Well, there's neurons and glial cells. This is the enteric nervous system that causes the gut to contract. And it's marked by both some PDX1 cells as well as a bunch of Wnt1 cells. Even more surprising, Irina looked down, way further down the gut tube in the ileum. So now we're a long way away from the pancreas and duodenum. And what she sees is, again, there's lots of Wnt1 cells in the external muscle layer, but there's still PDX1 cells that apparently have migrated down out of the pancreas and the duodenum in the embryo, down the gut tube, and are forming cells in the external muscle layer of the gut. And if you stain them, this is my favorite picture. So this is a whole mount of the embryonic gut tube stained with PDX1 Cre YFP. And what you see is this network of fibers that essentially is the enteric nervous system. It's marked by PDX1 cells. So PDX1 cells are forming at least part of the enteric nervous system surrounding the gut. And we know that they're neurons because we can co-label the PDX1 cells with MAP2 with neuronal markers. There's lots of neurons, though, that aren't marked with PDX1. Those are neural crest. So the majority of the enteric nervous system, the neurons in the enteric nervous system that cause gut contraction, are from the neural crest, but a significant minority are actually from a pancreatic lineage cell. And you can actually see them. So Irina looked at the Wnt1 Cre reporter animals and the PDX1 Cre reporter animals in the embryo. And she found that at 10.5 embryonic days, the neural crest starts to migrate down along the gut tube. About a day later, at 11.5, you see PDX1 Cre cells start to emerge from the duodenum and the pancreas and migrate down the gut tube about a day after the neural crest, but following the same tract down the gut tube and forming part of the enteric nervous system. So this is really shocking. This means that part of the enteric nervous system actually arises not from neural crest, but from an endoderm derivative from the pancreas and duodenum from endoderm cells. And the final thing that Irina showed is that not only do neurons from the pancreas and the duodenum migrate down the gut tube, but so do some stem cells. So she took the external muscle layer of the adult gut, dissociated into single cells, and see if she could grow sphere-forming cells. And the answer is she could. She grew two different types of sphere-forming cells. She grew small spheres, around 3,000 cells, and big spheres that came from this proliferation of single cells that are about 10,000 cells. And it turns out all of the small spheres, the vast majority of them, are marked by PDX1. So PDX1 gives rise to small stem cells that grow into small spheres in the external muscle layer of the gut. If you look at the big spheres, they're from the neural crest. They're marked by Wnt1 Cre, and the small spheres are not. So there's two different populations of stem cells, not only just neurons, but stem cells. One that comes from the neural crest and is lodged presumably somewhere in the external muscle layer, because that's where Irina dissected, and that gives rise to large colonies that are self-renewing and that make all the neural crest derivatives. They make neurons, they make fat cells, they make bone cells, and there's smaller stem cells that make smaller colonies, and these colonies, these pancreas-derived colonies, if you dissociate them in culture and differentiate them, will make neurons and all the cells of the pancreas. They'll make insulin-positive cells, glucagon, somatostatin, and acinar cells. So not only is the pancreas providing neurons that migrate down the gut tube and form part of the enteric nervous system, there are also a small number of pancreatic stem cells that have left the pancreas and duodenum and migrated down the gut tube and are present in the ileum as pancreatic stem cells that have the ability, at least in culture, to make not only neurons, but also insulin-positive cells, somatostatin, glucagon, and acinar cells. So what I've told you today is that a story that perhaps the pancreas evolved from the brain. And that story was based on the surprising finding that an endoderm-derived tissue, the pancreas, marked by PDX1 lineage, can actually give rise not only to all the cells in the pancreas, the hormone and the exocrine pancreas cells, but also to a small number of neurons. And we hint that that may be important for the evolution of the pancreas from the brain. Practically what this means is that there is a pancreatic stem cell present in the pancreas that can be isolated from adult mouse and human pancreas that can make insulin-positive beta cells, and we've also shown in work that Simon just published that those insulin-positive cells that come from adult mouse or human stem cells can actually rescue the high blood sugars of STZ-induced diabetic mice. So they seem to function as mature beta cells responding to blood glucose levels and secreting insulin to regularize blood sugar levels. Those stem cells are not only present in the pancreas, but they're also present in the external muscle layer of the gut where they've migrated embryologically out of the pancreas and duodenum down the gut tube. Most of the cells that are produced by the pancreas in the external muscle layer are neurons that form part of the enteric nervous system and we think may be important for gut contraction. Thanks very much for your attention.
Okay, we have time for some questions. What's wrong?
Well, thanks, Derek. So are these functional neurons? So do they have synaptic photons and demonstrate that?
So we haven't looked carefully at the EM level to know whether they're making synapses with each other. But that one whole mount picture I showed you where you could see the entire structure of the enteric nervous system makes me think that they're integrated, but we haven't studied them at the EM level, nor have we studied them functionally. One of the things that Irena's trying to ask right now is, can we selectively remove the neural crest-derived enteric nervous system versus the pancreas-derived enteric nervous system and see functionally is there a different effect on gut contraction? So we think may be able to do that. So that'll be an interesting functional way to get at that question.
Are these cells destroyed in diabetes, the diabetic who want to use cells?
Yeah. So one of the neat things that another postdoc in the lab, Rosita Ravi, has been doing is giving STZ, which is a drug that will kill insulin-positive beta cells in adult mice. And the way it gets in, the way STZ kills beta cells is it goes through the glucose transporter. And so you remember I told you the pancreatic stem cells themselves are insulin-positive but have a low glucose transporter. So you can use STZ and kill off 95% of all the insulin-positive beta cells and make the animals diabetic, but the pancreatic stem cells survive because they have low GLUT2 expression. And so it's interesting, when you isolate the stem cells from those diabetic animals, they're still there. There's not more of them, there's not less of them, but the surprising thing is, when they differentiate in culture from a diabetic animal, those stem cells now increase the number of cells that differentiate as beta cells, as if they're trying to make up for the loss or something like that.
I have two questions, short questions. The first one, have you found any easily positive neurons in the brain? First question. Second question is related to beta cells. As you may know that beta cell express GABA, which is the primary inhibitory neurotransmitter. So do you believe this beta cell is also kind of neuron?
So the second question is easy, and I'd say yes. So the fact that they're expressing a neurotransmitter, I think, is additional evidence that for the evolutionary history that of the pancreas from the brain. The first question is a harder question to answer. So just remind me the first question again. I forgot. It was so hard that I forgot the answer.
Have you found any easily positive
Oh yes. Yeah. So this turns out to be in the gut or in the brain. So this is, we haven't, and it's a huge controversy in the literature, about whether insulin is actually expressed in the adult mammalian brain. The evidence in the embryo is that there may be expression of insulin, but the evidence in the adult that there's actually insulin expression in the adult mammalian brain, I think, is very weak. And I don't think many people are convinced, and it's certainly an area of controversy. So, I mean, when I showed that the selection of species, the interesting thing is that in some species, the argument is that the original multicellular organism used insulin as a transmitter, and then it evolved insulin expression in the gut that became the pancreas. But if you look at existing species now, some of them have insulin in both pancreas and brain, some only in brain, some only in pancreas. So all the different derived functions or derived species are out there with yes or no for pancreas or for insulin in the pancreas and insulin in the brain. I hear a butt coming. No, I hear a butt coming.
Can you exclude that some rare neural crest cells express PDX1?
Yeah.
Or do you transgene?
It's a really great question. So the way we tried to ask that question, so Wnt1 is only expressed early in the neural crest, right? So the Wnt1 protein turns off as the cells start to migrate. But what we have done is at E10.5, as the Wnt1 lineage cells are migrating past, we've stained them for PDX1, and none of them stain for PDX1. So that at least says that at the time you might expect them to be passing nearby the pancreas, they're not expressing PDX1. And so, but it's a real hard question because all of these lineage tracing depends on the faithfulness of your Cree reporter, and it absolutely depends on that. Yeah.
There's this paper in Science, I think, basically alluded to the result that GABA was found in the pancreas like 20 years ago, something like that. Is there actual evidence coming to your evolutionary story? Is there evidence for this in the human?
I guess one piece of evidence is that in diabetics, because it's an autoimmune disease, you often can see in the blood antibodies against GAD and GAD67. And so the argument is, I don't know how good the evidence is that that's from, as beta cells are killed in the autoimmune disease, they release GABA synthesizing enzymes, which the immune system then generates antibodies to. So it's one of the things that often appears in the immune serum of diabetics is antibodies against GABA molecules.
Question, David?
Ask you to think using your gut. Do you have any indication whether you can derive the pancreatic, I guess they're not pancreatic anymore, from the gut in the human?
So we haven't done it, but it's a great idea. And we actually do think there's pancreatic still, because when we take them from the adult mouse smooth muscle layer around the gut, those cells are PDX1 lineage. They make endocrine cells in the dish, and they make neurons. So we actually think they are pancreatic stem cells. Whether we can do it from humans is another question, but we're going to try and do that experiment because we have now got human islet samples, and we know we can get pancreatic stem cells from them. So it's a great question that we're going to do.
Yeah, actually, we'll take one last question.
Wonderful presentation. Can you differentiate immunologically between those cells in the pancreas and those that are in the small bowel?
Right now, we don't have anything that differentiates between them. So it doesn't mean we can't do it, but so far, the neurons, for example, look very similar, but we haven't studied them in detail. We know they express general neuronal markers, but whether they express the same transmitters, things like that, we don't know. And whether they have different molecules on their surface, we just don't know.
Do you believe they're likely to be different?
I think they're from the same lineage, so I would say maybe not, right?
So the question is then in
Yeah. Diabetics.
diabetics.
Yeah, it's a good question. So would those cells be killed? Yeah. Well, if you think that the autoimmune system is only is only a reaction against the insulin positive cells, so you don't, the autoimmune disease does kill some glucagon and somatostatin, doesn't kill other cells in the pancreas, it kills the beta cells. And if the pancreatic derived cells in the gut have become neurons, then maybe they won't express the cell surface markers that the diabetic autoimmune disease is attacking. So maybe they'll be spared, I guess.
Okay. Well, people would join me in thanking Dr. Vanderkooy for a well-designed.